Okay, so in the next 20 minutes, my goal is to try to summarize all of the therapies you're going to hear about over the course of the next 2 days and all of the handouts in your packet of clinical trials and preclinical drug targets to put them in context of the molecular and cellular events that John talked about a minute ago so that you understand what categories each of the drugs is targeting. So as you all know, Duchenne is caused by the absence of the dystrophin protein, which in this cartoon of a muscle membrane is the blue molecule, which at one end of the dystrophin protein binds to the skeleton inside of the cell, and at the other side of the protein binds to a group of proteins in the membrane called the glycoprotein complex that in turn bind to proteins outside of the muscle cell. And so dystrophin provides a link between proteins inside and outside of the cell. When dystrophin is missing, the entire complex of proteins at the membrane becomes destabilized and are no longer present, and you lose the connection to stabilize the membrane by making the connections between proteins inside of the cell and outside of the cell. This leads to muscle membranes that are susceptible to damage. When the muscle membrane's damaged, it looks like a single muscle fiber that doesn't have dystrophin, when it becomes to have membrane damage, the normal striated pattern that is present in normal muscle becomes disrupted, and this membrane becomes leaky. And so what happens is that proteins from the inside of the cell leak out of the cell into the bloodstream, and likewise proteins from the bloodstream leak into the muscle, causing an imbalance in numerous molecules that are typically present inside the cell. And so this damage can also be seen in a cross-section, where in this panel the fibers are coming out of the plane of the screen at you, and each of the green muscle fibers are those that actually have blood inside the muscle fibers and have leaked out the contents that should normally be present in the muscle. So these cellular events can basically be broken down into a few broad categories. And so in skeletal muscle, these categories can be broken down into muscle weakness that occurs before membrane damage. And when the membrane is damaged and a fiber starts to die, immune cells cause inflammation and come in and clear up the area. And these immune cells can have both positive and negative effects. The positive effects are that they clean up the dead tissue and allow regeneration to occur. So skeletal muscle is able to regenerate even in Duchenne muscle for many, many years. And so there's a large regenerative capacity, but eventually this regenerative capacity wears out, and whenever a muscle fiber dies, it becomes replaced by connective tissue or fat tissue, and this is what prevents the muscles from working correctly. In the heart, these steps are very similar, in that first you get muscle weakness before there's any membrane damage, and then there's membrane damage. The role of inflammation in the heart is less well characterized, and this is partially because, unfortunately, there is no regeneration in the heart. So as a cell dies, it cannot regenerate, and it becomes immediately replaced by connective tissue and sometimes fat. And this is an MRI of a 14-year-old DMD boy of the heart, and you can see this patch of damage. And so one point that I want to make is that all of these steps occur while the muscles are still functioning properly, while skeletal muscle is still functioning properly, when your sons can still walk and run and do everything. And the same with the heart. All of these steps occur while the heart is still 100% functioning at the level of the organ. And so the good news about this is that there's actually a very long therapeutic window to treat DMD. So again, I'm going to try to quickly go through everything using the names that are in your program of the titles of the talks and using the names of the drugs that are in your handouts. And so for each step that we just talked about, I'll address the step. And we'll talk about the drugs or the therapies that are targeted towards each step. And the name of the drug will be bolded, and that's either in your itinerary, in your fact sheets or in the schedule. And anything that's not yet in clinic, a preclinical drug, will be italicized. And where the target is known will be before the drug in non-bolded print. So, from when the gene was cloned, the most logical approach to therapy was to put back dystrophin. If we can get 100% replacement of a full-length dystrophin, we would be able to cure DMD. But this is not as simple as it sounds. For many years, people investigated many delivery systems to try to deliver full-length dystrophin back. And it's the one that's the safest and the most efficacious is a very small delivery vehicle, and so it can't hold full-length dystrophin. And so there's a gene transfer clinical trial of microdystrophin, but this is a dystrophin that leads to Becker muscular dystrophy. So although it would improve muscle function, it would not improve it back to normal. It's similar. The similar approaches to restore dystrophin that are only possible for some mutations are exon skipping, and these drugs are listed here. There are 3 different trials or therapies in development or in clinical trials right now that are all involve skipping exons and again making a truncated dystrophin back in skeletal muscle. A third approach is for patients who have stop codons, is to be able to read through the stop codon as if it wasn't there. And this actually would make a full-length dystrophin with just a single typo in it. And so these are all therapies to actually put back dystrophin at the molecular level. And there's a fourth approach, which is to try to put back cells, muscle cells from normal donors to make new muscle that has dystrophin. And there's a myoblast transplantation trial in planning, and there's preclinical studies on using induced pluripotent stem cell therapies. So a caveat about restoring dystrophin is that kids without dystrophin may see it as a foreign antigen, as something invading their bodies. And we don't know yet what the long-term immune responses might be. And so many labs have taken approaches of looking at upregulating compensatory linkages, other ways to protect the membrane by connecting the inside and the outside of the muscle cell. And so the closest protein that's similar to dystrophin is called utrophin, And if you can overexpress utrophin, it can take the place of dystrophin, and it's already present in skeletal muscle of boys with DMD, and so it would not be seen as a foreign antigen. And SMTC1100 is one drug under development in clinical trial planning that would upregulate the utrophin that's already there and possibly compensate for dystrophin. Another molecule in preclinical development is called biglycan, and that's the orange protein here that actually helps stabilize this group of proteins that can bind to dystrophin and will upregulate utrophin and again stabilize these utrophin complexes to reconnect the inside and the outside of the cell. And then a third compensatory linkage approach is to upregulate other complexes unrelated to dystrophin and utrophin that are called integrins laminins that separately provide connections from the inside to the outside of the cell. So those are still there in Duchenne muscle, but if you can, again, overexpress them to help compensate for the loss of the dystrophin connection, that may also provide a therapeutic approach. So there are other ways besides restoring dystrophin or compensatory linkages that may target membrane damage. There's a clinical trial being planned on GAL-GT2 gene therapy. And what GAL-GT2 does is it puts sugars on the proteins of the complex that associates with dystrophin and helps stabilize that at the membranes. And the sugars help coat the membrane and make it stronger. Another approach that's similar is called Carmacil, and this is a molecular Band-Aid that is known for many tissue types to be able to just non-specifically seal the membrane. There's a class of drugs called mineralocorticoid receptor antagonists that have been used in both pediatric and adult heart disease for decades, safely and effectively. And these were thought of as targeting fibrosis, but our recent clinical trial data as well as preclinical studies show that they actually also help muscle weakness and membrane damage. And so there's been one successful clinical trial on Duchenne patients with cardiac outcomes, and allopurinol was shown to actually slow down the progression of cardiomyopathy in DMD boys. And there's a second clinical trial that's recruiting patients right now, comparing spironolactone and allopurinol of which one might be most effective in DMD patients. But again, these are drugs that are used in many patient populations. And my lab is actually right now conducting a preclinical study because we found that these drugs had the same benefit for skeletal muscle in mice, and we're again optimizing which drug is better to be able to move this into a clinical trial. So there are some other groups of targets that will target muscle weakness. And so one of these is myostatin inhibition, and there are actually 3 trials listed in your handout that target myostatin, and inhibiting myostatin causes growth of muscle, so to be able to compensate for the loss of muscle in Duchenne. And so one of these is a gene transfer follistatin, and one is a drug from Pfizer, an antibody against myostatin, and one is an antibody against myostatin from Bristol Myers Squibb, and the numbers are too long to name. And another approach to increasing muscle strength is targeting the androgen receptor, and there's DT200 that's in development, and then I guess was released this morning, potentially a successful trial, though I saw the email and haven't read the data yet, for HT100, which also targets muscle weakness and maybe inflammation and maybe fibrosis, but I'm not sure the mechanism's entirely known, so I didn't list a target. So there's another group of therapies that you're probably most familiar with that target as a primary endpoint target inflammation, but appear to have secondary effects on improving muscle strength and preventing muscle weakness. And so the most well-known of these are glucocorticoids, prednisone and deflazacort, and there are several trials going on with these to optimize dosage and timing and compare deflazacort and prednisone. There are also a new glucocorticoid is under development, VBP15, by one of our speakers later in the session, to have the same benefit of glucocorticoids without the side effects. Other anti-inflammatory targets are NF-kappa B, and NF-kappa B is one of the signaling molecules that goes a little out of whack when you have membrane damage. But NF-kappa B inhibition also not only has an effect on inflammation, but directly targets muscle and appears to improve muscle strength. And so there are two different approaches in clinical trial or preclinical studies that target this pathway. There are two other anti-inflammatory approaches, a combination of ibuprofen and isosorbate dinitrate, or isofen, and inhibition of the TLR receptor IMO 8400. Okay, so again, when the muscle membrane is damaged, the balance of many molecules in the cell as a secondary effect is disturbed, and so there are a number of therapies in development or in clinical trial that target some of these downstream mechanisms that again may have some therapeutic effects particularly on muscle weakness. So Katina Raxone targets the secondary effects on mitochondrial function, epicatinin, Ricol and AT300 target calcium handling, PD5 inhibition to tadalafil target the neuronal nitric oxide synthase that's not localized to the right place, and there's another trial with using tamoxifen. So just to summarize and sort of end with the possibilities of the therapies that really may become realities over the next few years. Again, ideally, it would be great if we could put back 100% dystrophin, full-length dystrophin, and completely replace all of the dystrophin that's missing. And that would stop muscle weakness, stop membrane damage, stop inflammation, and stop fibrosis, and be an effective treatment of all skeletal muscles and the heart in DMD. Similarly, while the kinks and the bugs and the problems with getting gene therapy of a really big protein like dystrophin are being worked out, which may take a long time, there's the possibility for combinatorial therapies to use microdystrophin and exon skipping that will partially restore part of dystrophin, and combine it with therapies that target membrane damage and therapies that target inflammation inflammation and therapies that target fibrosis in a combinatorial fashion. And again, if you stop the therapeutic window is large, and if you can slow down or stop any of the steps in this pathway, you will likewise wind up with an effective treatment of skeletal muscle and heart. And if that idea seems far-fetched, I just want to remind you of how we treat cancer and heart disease and HIV. All of those are treated and managed for decades by combinations of drugs. Anyone who has heart disease is probably on 2 or 3 or 4 different drugs, and they are clinically managed for sometimes decades and decades. And the same with cancer. Chemotherapies are usually a cocktail of drugs that target cancer. So, I think we really need to start thinking about combinatorial therapies and how we can best target multiple pathways in the disease to get to that 100% efficacious point. And that is the end. And we're waiting for questions till the end, right?